If you're driving by a cornfield at 60 miles an hour or something like that, you're probably shaking your head and saying, I don't see any variation in a cornfield. But you realize that after you spend a few weeks out in the cornfield, there's a tremendous amount of variation between one corn variety and another corn variety. So the type of work we do out at the Musgrave Research Center is essentially we're trying to figure out how natural changes in DNA allow a plant to grow in different environments and produce different phenotypes.
You'll hear everybody say the word phenotype, and what that really just means is a physical attribute. So a phenotype in a person could be blue eyes and brown hair. A phenotype in corn could be how tall it is, could be when it flowers, could be the color of it. Any physical representation that we can measure is a phenotype.
So some of the things that we're measuring are things you can see with your eye. But sometimes what we call the phenotype is something that the plant's doing on the inside, okay? So sometimes we'll be grinding up that corn plant or the leaf and asking how much sugar is in there, how much starch is in there, how much protein is present. And so essentially we're down at the level of we're measuring various molecules that are inside the corn plant. And so all of those are what we consider the phenotype. The second thing we need to measure, if we're going to make that connection between the phenotype and what we call the genotype is the DNA. And so maize has a genome with 2.3 billion base pairs. And so, I mean, this is always still an extraordinary number to me, that first off, every cell contains 2 copies of this 2.3 billion base pairs. But if you were to take that DNA and actually stretch it all out, it would be 1.5 meters in length per cell. But all that DNA gets jammed into one little cell that you can't even see. But we now have technology. These DNA sequencers have been one of the major revolutions in genetics in the last 20-some years, that it's now possible for us to sequence the genome of any species in a day or 2. So we can sequence trillions of base pairs every week using the modern DNA sequencers and allow us to identify base pair by base pair what's going on within these genomes.
So I guess what I do day to day is we generate really large data sets, either about the genotypes of plants or the phenotypes of plants. So that means either what's in their DNA or what they look like in the field. So we collect large amounts of data by doing sequencing, as people have probably heard about, relative to disease, or measuring a lot of plants in the field. And by combining these two, we can figure out the DNA sequence that's responsible for specific things that we see in the field, phenotypes, for instance. So it's a mixture of being out in the field, being in the lab, and I guess what I do most of all is at the computer to marry these two data sets together. I think of my work really as being at the center of a funnel. So we have these great diversity of corn, which is like this big funnel like this, and then we try to use different biological filters to just get down to the stuff that matters.
So once we've done the math to figure out these are the base pairs that matter the most in adapting a corn plant to a particular environment, what we then do is we start making crosses.
The other thing we are actually doing in the field is crossing plants, so essentially controlled reproduction. We're deciding what 2 things mate together.
And we can then look at the offspring from that cross, and we can make a decision really quickly to say, keep that corn variety, are we going in the right direction or not? And so we can then grow out make thousands of different seeds, and we can say, these are the 10 seed that are most likely to work in this environment. And we keep those and we make new varieties based on that genetic knowledge.
By having slightly more accurate predictions about how well plants will do, we'll be able to increase the speed that we breed plants, or that we're able to breed plants for specific environments. Like, let's say we know that specific genes are important for calibrating your ability to tolerate drought, or a plant's ability to tolerate drought. So if we could just figure out the genes that matter for drought and then make predictions based on those, we could potentially be more accurate and breed for more specific environments across a greater portion of the world.
So, for example, one of the earliest studies we did is we looked at how natural variation produced variation in beta-carotene content. So people in large parts of Africa, they rely on maize for a lot of their food throughout much of the year, and they get a lot of their vitamins from maize. But maize doesn't have that much beta-carotene content, and so hundreds of thousands of children a year essentially go blind because they don't get enough beta-carotene. But then there's actually several 100 million people around the year that if they don't get enough beta-carotene are immune, their immune system is compromised. And so they get sicker more often. They have all sorts of other problems with their immune system not being as robust as it should be. And so we've studied out at the Musgrave Farm how maize varied for provitamin A content, or this beta-carotene content, and we're able to figure out 2 genes and 2 natural variants of each of these genes that allowed us to increase beta-carotene content about 15-fold. So that now has resulted in essentially new varieties of maize that are being sold in Zambia that have 16-fold more beta-carotene and are providing better nutrition for children all across Zambia. So the exciting thing about genetics and where we are right now is combining our knowledge of genomics and how things grow out in the field is that not only is our group doing this, but hundreds of groups around the country and around the world now have the technology within their hand to tap natural variation that has been out there and really combine it together to create new varieties of maize and new crops that can really adapt to societies and the world's needs.